and thank you for attending today's webinar in GenScript's 2020 Spring Webinar Series. I am Dr. Samantha Zaroff, a Senior Marketing Specialist here at GenScript, working with our Neoantigen peptide synthesis services. This afternoon, we have the great pleasure to listen to Dr. Raymond Miller, the Senior Global Product Manager of Molecular Diagnostics and Therapeutic Materials at GenScript. Today, Dr. Miller will be presenting his webinar about using a predictive bioinformatics algorithm for difficult neoantigen peptide synthesis for precision immuno-oncology therapeutic development. In order for this webinar to run as smoothly as possible, we ask that you please mute your personal microphones and type all questions directly into the chat box throughout the presentation. At the conclusion of the presentation, we will host a formal question and answer session to directly address your comments. Now, without further ado, it is my great pleasure to turn today's presentation over to our speaker. Dr. Miller, the floor is all yours.
Awesome. Thanks, Sam. Thanks for the kind introductions, and thanks to everyone that took time out of their day to hear today's webinar. So as everyone can see from the title slide, we're going to be talking about predictive bioinformatic algorithms and neoantigen peptide synthesis. So before we kind of dive into the details and talk about how GenScript is helping to reliably synthesize these very difficult peptides, I thought it'd be good to start with kind of a big picture, to think about why neoantigens are important and how they're being used and can be used in so-called cancer immunotherapies. And so we're going to start with a light introduction here, talk about immunotherapy. We'll then transition over to neoantigens and how they're potentially being used in the development of new personalized treatments. We'll then shift gears a little bit and talk about how peptides are being used as part of the development of these various neoantigen-based therapeutic platforms. And then last but certainly not least, we'll describe a little bit about the work that our team has done and how we're working to improve the ability to synthesize these very difficult peptides. So without further ado, let's get started. So I think it's fair to say, and I think the audience would agree, that in general, traditional treatments like chemotherapy tend to have a lot of various drawbacks when treating cancers. Obviously, they're still the frontline treatments and the preferred treatment, but there are various side effects. And so there's a lot of interest and a lot of energy being spent on immunotherapies. Of course, in concept, immunotherapies are very, very easy to understand. The idea is a human being, a patient, should be able to have an immune system that responds well to a cancer and attacks that cancer and destroys it. However, what often happens is due to a variety of conditions, over time, the patient will end up having a less than vigorous immune response. And so immunotherapy is really about getting a patient's own natural defense system to be more active or more readily able to respond to a particular cancer. And in general, there are two broad buckets for immunotherapies: checkpoint inhibitors and T-cell therapies. Checkpoint inhibitors are really all about taking the brakes off the immune system, really helping the immune system ramp up, be more active in the hope that it's able to recognize the cancer cells within an individual. And obviously, there's a lot of exciting work that's been done. There's a lot of treatments on market today, but overall, the efficacy of these treatments is not perfect. And we'll talk about ways that folks are trying to mitigate that in a second. T-cell therapies are a slightly different approach. Instead of just ramping up non-specifically the immune system, T-cell therapies are all about taking the patient's T-cells or from a healthy donor and then modifying or training these T-cells such that they're more readily able to identify particular cancer antigens. And so you extract cells from the patient, you then go ahead and culture them ex vivo, and you modify them by having them express a CAR or TCR. So then you go ahead and take the cells, put them back into the individual, and have them go and hunt down cancer cells. So again, both approaches have their pros and cons. Like everything in life, nothing is perfect. And in general, it's fair to say, though, that all the results are promising, efficacy is not where we'd like it to be at this point. And so this is leading to a variety of strategies. Of course, combinational treatments are becoming very, very popular right now and are going through clinical trials. But additionally, there's been a lot of focus around so-called precision medicine. So when I think about precision medicine, I really think about it in terms of two very broad categories. One is taking existing off-the-shelf population-centric medicines and being more effective at determining which patient will get which medicine. So if we start with the patient, understand their specific cancer biology through tumor profiling, for example, using next generation sequencing, we can identify variants that or abnormalities that we know are associated with positive clinical outcomes if a given treatment is given. And then we give the patient that treatment. So for example, checkpoint inhibitors. There's an increasing body of research being done to indicate that the more mutations or the more variants that are present within a tumor, the more likely a patient is to respond positively to checkpoint inhibitors. And so this is a way that we can be more efficient at providing treatments to different patients. Obviously, gene fusions are a hallmark of cancer, and they can be treated by things like tyrosine kinase inhibitors. So that's one approach, is take a medicine that's already off the shelf and just be more effective at determining who gets it and when they would get it, and what the dosage should be. The other approach is a little more germane to our conversation, which is really truly personalized therapeutics. And it's worth pointing out at this point that this is really a very rapidly evolving field. And obviously there's a lot of unknowns, but in general, the concept is straightforward, which is, again, start with the patient, do tumor profiling, understand their cancer biology a little bit better, and then identify potential targets that could be or could respond to therapies. So drugs, treatments. All right, great. So what kind of drugs, what kind of treatments are we talking about? And again, I want to emphasize that we should think about the biomarkers that are involved in the biology of a given patient's cancer. So what are the targets we should look for, and then what are the treatments? And the targets that folks are, I think, really excited about, of course, are neoantigens. And in general, neoantigens are antigens that are variants, indels, sometimes frameshift mutations that are found in cancer cells, but not found in healthy cells. And this is really important because a lot of the treatments that are on market today or under development focus a lot on cancer-specific antigens, and often these are self-antigens. And so this can lead to, unfortunately, some potential cytotoxicity challenges. It's very much expected that neoantigens are going to be highly individualistic. So every patient is going to be a little bit different. There are obviously hundreds and potentially thousands of variants present within a patient, but only a handful are going to be held in common between different patients. So again, truly personalized treatments. If true, neoantigens are expressed, they're also presented on the cells, and presented in such a way that the immune system could, in theory, see them, find them, and attack them. And given the fact that neoantigens are, again, by definition, not self-antigens, they're likely to be highly immunogenic. It's obviously a well-known fact that self-antigens are greatly not preferred by the immune system and selected against for obvious reasons. So we've identified a potential area of biomarkers that could be used in a personalized immunotherapy. How are they being deployed? So in general, the two approaches that are under active research and development today are neoantigen T-cell therapies, or personalized T-cell therapies, and personalized cancer vaccines. So T-cell therapies using neoantigens are very similar to kind of the off-the-shelf T-cell treatments like Kymriah. You essentially, again, they'll start with the patient, identify neoantigens, and then you go ahead and you try to insert into the patient's T-cells receptors that respond or recognize these specific neoantigens. So again, very, very, very highly personalized. The second approach is slightly different. Instead of using cell-based approach, you challenge the immune system with a nucleic acid or with peptides that encode for a neoantigen in the hopes that this will prompt the immune system to recognize this as foreign, and then go ahead and then mount a response, and then ultimately to hunt down and find the cancer cells that express these neoantigens. It's worth, again, emphasizing that these treatments are under development, and in the case of personalized cancer vaccines, we have some clinical trials underway, so obviously a lot of this is to be proven and the efficacy to be established. But in general, again, a very, very exciting area of research and development in the immunotherapy space. So it's worth thinking about, well, how are these platforms going to be developed? What's the process going to look like? And what I'm showing you on the top of this slide is kind of an idealized process. So again, start with the patient, perform a biopsy, extract nucleic acid, and then go through a round of initial neoantigen identification. So again, whole exome sequencing, RNA sequencing, using in silico methods to try to rank out and try to identify potential neoantigens. It's worth pointing out that obviously, even with all these efforts, it's not a guarantee that the outcome is going to be a really true neoantigen that is ultimately going to allow the immune system to invoke an immune response or to recognize and invoke an immune response. And so often what's required is the production of peptides. And these peptides themselves are then used to confirm the possibility that these are real neoantigens that could respond, that could invoke an immune response. And you do this through a variety of assays like ELISpots and of course flow cytometry. So you're doing some sort of preclinical screening to say, okay, are these real neoantigens? And can the immune system recognize them and mount a response? The next phase in this idealized process is, of course, you want to test the safety and toxicity of this platform. You'll do animal testing in rodents and then perhaps in non-human primates to establish the safety of the platform, and then ultimately go into clinical testing, enroll a couple of dozen patients and try to work with them to help put their cancer into remission. And obviously, it's again worth pointing out that these clinical trials are ongoing, but afterwards, of course, we want to determine were we able to help patients, and was the help that we provided what really ended up causing their cancer to go into remission. So peptides are a critical component for this workflow, and they're used really in three major stages. The first one is kind of the initial screening, whether it's in early R&D or whether it's during a clinical trial and you're working with a specific patient. There's a lot of mapping of T-cell receptors and particular neoantigens to determine, again, is there immunogenicity, is there a response? For researchers developing personalized cancer vaccine platforms that are peptide-based, obviously there's a requirement for peptides that encode those potential neoantigens to be made and then ultimately put into a patient to try to invoke an immune response in the patient. And then last but certainly not least, peptides are used for determining efficacy. So determining, okay, did your treatment have a positive impact and why was that the case? And so again, peptides are critical for this. But in general, what we found is that peptides are really the bottleneck in this workflow. I think it's fair to say that sequencing has become more standardized. Obviously, in silico algorithms continue to be improved. ELISpots, flow cytometry have been around for decades. Really, the challenging part is getting a reliable source of peptides as quickly and efficiently as possible with a high success rate. And so we know, again, from our own experience, from the experience from our customers, that this can be a pain point and this can be a bottleneck and take a lot of time. And it's worth thinking a little bit about, well, how are peptides made? And then how can we improve the situation? So in general, peptides are synthesized through one of two ways. There's either chemical synthesis or there's recombinant expression. I would say the vast majority of peptides being synthesized today on market occur through chemical synthesis. It's obviously a more standardized process. There's a lot more control over that process. And of course, there's also, since it's being done kind of in an artificial setting, there's less of a chance of certain toxicities or certain impurities that come along for the ride. So in general, synthetic production is being used, and that can be broken down into a variety of different factors. It's worth talking a little bit about the actual peptide synthesis, where it's building a chain by going through a standard chemistry that's been first described as a liquid phase chemistry, and doing a coupling of the carboxy group of incoming amino acids, the N-terminus of a growing peptide chain, and then ultimately removing this protective group over and over again until you have a full-length peptide. That's the basics, but in general, there are three types of artificial chemical peptide synthesis: liquid, solid, and microwave. Each approach has their own pluses and minuses. In general, we found that microwave technology tends to be the best approach, although GenScript has all three platforms available. We found that in our own research that microwave technology is best at a fast turnaround time. and then it ultimately works really, really well for peptides that are less than 50 amino acids. And so for most of our customers, we are synthesizing peptides at most up to about 30, 35 amino acids. So the platform works really, really well, and it overall has a pretty high success rate. But that's not to say that these other platforms don't have particular value, and we ultimately will turn to these platforms depending on the project that we take on. So that's how we do it. The why, though, is why is it so difficult for folks to synthesize these peptides? And we know that this is a common problem across the market, and we know that this is, again, leads to frustration. And in general, the reason why it's so difficult to synthesize these peptides is a variety of factors involving the intrinsic characteristics of peptides. So length, charge, hydrophobicity, the amount of yield that's required, and the purity that's required. So in general, peptides that are over 20 amino acids tend to become more and more difficult to synthesize. Charge can have a huge impact on peptide synthesis difficulty. The further from neutral charge is of a peptide, the more difficult it's likely to be. Same with hydrophobicity. The more hydrophobic a peptide is, the more difficult it's going to be to synthesize, and the more insoluble it's going to become, the more likely it's going to aggregate. And obviously that's not something that we want to happen, and it's obviously something that researchers don't want to see as well. Yield is also an issue, although it's a little bit of a lesser issue. Milligram amounts typically tend to be effective for most assays. Purity, though, becomes a bit of a challenge. It's very, very difficult to purify these peptides, largely because of the charge and the hydrophobicity, causing issues with solubility, and then also, as I mentioned before, aggregation can occur. So again, very, very difficult challenges. So how do you overcome them? Well, at GenScript, we do it through a combination of different approaches. Ultimately, what we end up doing is we use, yes, experience, the multiple platforms that we have, and the refinements that our team has done in making those platforms even more effective and fast. But we also are very, very active in algorithm development. And we're very active in trying to, using the information that we have from our clients, choose the best course for every single project. And so we focus first on assessing the peptide through our NeoPreTM algorithm. So this is our proprietary internal algorithm, and this tells us a little about the peptide. Obviously, we'll know the length, we'll know the hydrophobicity score, we'll know all these things. And based on our mining of our own internal data, we know the challenge this peptide is likely to have for the various platforms that we have. And then we can choose the best course of action. We can pick the right synthesis platform to maximize success and to achieve success at a fast turnaround time. So again, this is the general approach that we're taking, which is when we take a pilot project on, we engage with our customers. We then go through and establish the project goals and then have a rough plan of action. We receive their sequences, we run them through our algorithms, and we use this as a starting point to outline the best process to move forward and to, again, get a reliable amount of peptides quickly and efficiently. So our algorithm is currently available online, and it's a very simple algorithm. It's really just go ahead, put sequence in, submit sequence, and then ultimately gives us an idea of, again, the overall peptide synthesis difficulty and what is the appropriate response, what's the appropriate action, what's the appropriate process. Obviously, the algorithm that we put out is only a partial algorithm, and a lot more of the details are hidden internally. So at this point, our algorithm, again, does its analysis and it comes out with scores. It tells us, okay, this is difficult based on these physical characteristics. And if so, what is the default recommended protocol that we should use? How should we approach things? And again, this is going to vary on a lot of factors. So it's not just the peptide sequence itself, but how much material you want, what purity you would like. And this, again, will all factor into how our system determines what's the best course of action. So let's look at some specific numbers. So what I'm showing you right now is I'm showing you a batch of peptides that we produced, I believe in the course of a couple of projects. So this is kind of synthesized data off of several different runs. And what we wanted to do in building this algorithm is really go ahead and really understand how effective the algorithm was, how accurate it was. And so what we ended up doing is going ahead and then taking this algorithm, developing the peptides, and then ultimately backtracking and saying, okay, based on our results, was the algorithm able to determine if peptide was easy or difficult? In general, what we found is that honestly, most of the peptides that we get tend to be on the easy side, although we have a fair number that are difficult as well. In this case, we biased our sample such that we had about even amounts of both easy and difficult to synthesize peptides. Overall, our accuracy is about 80%, a little greater than 80%, especially for difficult peptides, which is great because this obviously informs our ability to make these peptides and make them again as efficiently and reliably as humanly possible. So here's another study. We're looking at a different period of time. I believe this was over a 2 to 3 month period, where ultimately we were synthesizing over 2,000 peptides. And again, this is a variety of different projects, all of them being neoantigen focused. Ultimately, we had an incredibly high success rate of 95% in delivering peptides. And we find that this is definitely, well above industry average. In general, again, for this particular time period, we had a nice mixture of difficult and easy peptides, slightly more easy peptides for this 3 month period. Overall, we had most of our peptides that we synthesize for neoantigen work tend to be about 8 to 19 amino acids. 15 more overlapping libraries tend to be often very, very common. We also, of course, do synthesize longer peptides greater than 20 amino acids. So, for example, peptides that are 33 amino acids in length tend to be very, very popular. Overall, the results are pretty much, in line with our expectations, and again, well above market average, which is overall we had a 95% success rate. But especially for short peptides, using our NeoPre algorithm to guide us in terms of what's the best synthesis platform, we ended up with 98% success rate. We also had a great turnaround time of overall about 10 days. This is, I think, a phenomenal turnaround time. Often when we're engaging with prospective clients and trying to get a better understanding of how we fit in the market and their expectations, we get turnaround times of anywhere from 4 weeks to 6 weeks to even sometimes 8 weeks to never on the market. And so to be able to do this in 10 days is impressive and really, I think, shows the hard work our team has put into this platform. Even for longer peptides, we still overall have a pretty good success rate. It's 85%. Obviously, there are areas of improvement that we want to investigate. We're looking into new synthesis platforms, then obviously tweaking and improving our own platforms. But using our NeoPre algorithm, along with our expertise, along with all the tools that we have in our toolbox, we're still able to, again, get an 85% success rate. And that overall, our turnaround time, although it's not as great as we'd like, is still a very, very, very good 19 days, which is, again, well above market expectations. So this has really led us to believe that the platform, the service that we offer, which again is highly customized, is going to meet the vast majority of projects involving neoantigen therapeutic research and development. Switching gears slightly, I want to focus a little more on customization, and then we'll do a quick wrap up. So in general, we have a wide range of tools in our toolbox. We can not only synthesize these very difficult peptides reliably and achieve an overall 95% success rate, but we're able to offer a variety of different QC options. It's worth pointing out that our peptides, of course, are research use only or research grade peptides. We are actively working to develop GMP grade peptides. And as part of that, we obviously want to offer a wide range of different QC tests. So for example, removing endotoxin, we guarantee that. Removing our counter ion, so we use TFA, making sure that that's at a very, very low, almost infinitesimal percentage is important. Making sure that our peptides are soluble, that they obviously can go into the solution is obviously important. But there's a wide range of tests that we do that ultimately will go into the GMP process that we're developing today. And again, it's worth emphasizing, even for research use only peptides, we're able to offer all of these services or a certain combination. Depends really on our client's specifications, your requirements essentially. Additionally, what we're able to do is we're able to take things the next step further. So often what we hear from our clients is there's a lot of interest, especially in early screening, especially in the development of a platform, to really understand the interaction between TCR and the antigen. So there's often a lot of a need to really understand what the critical residues are and do a lot of really great mutational libraries. So we offer a range of different libraries, alanine scanning, overlapping, and then T-cell truncation libraries, which are obviously for this service very, very popular. So these are just, again, some of the options that we bring to bear. So, summary. In general, neoantigens are an exciting area of personalized therapeutic development. Obviously, there's a lot that needs to be done and a lot of exciting work that will be done to show their clinical efficacy, but they have a lot of potential. They are potentially a source of or a target for the development of these personalized treatments. Neoantigens are often found in only cancerous tissue, but not within wild type tissue. Overall, they're thought to be more immunogenic, and that overall they're being developed or deployed in 2 different platforms, personalized cancer vaccines, and then also neoantigen T-cell receptors. What GenScript does is we provide the peptides that are used in research and development and clinical efficacy, and then ultimately our ambition is to offer peptides that could be used for a peptide-based personalized cancer vaccine. We are able to reliably synthesize these peptides with up to 95% success using a variety of tools, platforms, experiences, and also our NeoPreTM bioinformatic algorithms. And using all these factors and a very customized project-based approach, we're able to go ahead and to get you the peptides you need with a high success rate and a fast turnaround time. Overall, we feel that we're a very, very well-established provider in this space. We've been doing this for 2+ years at this point for a variety of different customers in academia and industry. And we continue to expand and we continue to work to improve the platforms that we have to offer. And so with that, I want to thank everyone again for their time. And I want to open up the conversation to questions. And I look forward to the interaction. Thanks very much.